Imagine a city bustling with billions of inhabitants, where every single resident knows exactly what to do, when to do it, and how to coordinate with everyone else. And they do this all without uttering a single word. Sounds almost impossible, right? Well, something pretty similar is actually happening inside your body right now, every second. Trillions of cells engaged in this silent, incredibly intricate dance of communication. Today, we're diving deep into cell signaling by chemical messengers, and we're drawing our insights from a really foundational text, Marks Basic Medical Biochemistry. Our exploration is going to uncover how your cells talk to each other, how they integrate these complex functions, and adapt to, the ever-changing conditions of life. Our mission here is to sort of demystify the cellular language for you, to reveal the who, what, and how of these really critical biological conversations. By the end, hopefully, you'll not only understand the surprising elegance of this system, but also why grasping these fundamental mechanisms is absolutely key to understanding health and disease. Think of it as your shortcut to being genuinely well-informed about the language of life itself. So where do we even begin with cells talking to each other? It sounds incredibly complex, but I suspect there's a beautiful logic at its core somewhere.
Oh, there absolutely is. A profound logic, actually. We really have to start with the messengers themselves. When you zoom out a bit, all complex organisms, I mean, all of them, depend on this communication just to survive. It allows for specialized functions across different organs and tissues, making sure the body operates as, well, one cohesive unit. And what's truly remarkable, I think, is the sheer variety of these messengers and the clever strategies they use to get their information across. These chemical messengers, sometimes called signaling molecules, they're basically the words or signals in our cell's vocabulary. So a cell secretes one in response to a specific trigger. It then travels to a target cell, binds to a specialized listening protein, that's the receptor, and then without being used up itself, it triggers a response inside that target cell.
Okay, so it's not just one kind of message flying around. It sounds more like a whole cellular post office system, with different delivery speeds and specialized mail types.
That's a great analogy, actually. And you mentioned delivery methods. Distance really matters here. For endocrine signaling, think long distance. It's like broadcasting a message across the entire body. Messengers, like the hormone insulin, for example, are released straight into the bloodstream and travel to potentially very distant target cells.
Okay, like a national broadcast. What about closer communication?
Right. Then you have paracrine communication that's much more local, like neighbors talking over the fence. Messengers just travel between nearby cells, often just diffusing across a tiny gap. Neurotransmitters like acetylcholine or ACC at a synapse, that gap between a nerve and a muscle, they're a perfect example. They only really hit the receptors right across that little gap.
And I think I read somewhere that cells can even talk to themselves. Is that right?
Absolutely. That's autocrine signaling. A cell releases a messenger, and that messenger acts on the very same cell that made it, or maybe on adjacent cells of the exact same type. It's a really efficient way for a cell to kind of fine-tune its own behavior.
Self-regulation built right in.
Exactly. And beyond just the travel distances, we also tend to categorize these messengers into big families based on their main jobs. The nervous system, for instance, relies heavily on neurotransmitters, think epinephrine. The endocrine system uses hormones, insulin, glucagon, cortisol are big ones. And your immune system has its own language using cytokines, things like interleukins and tumor necrosis factors, which are crucial for coordinating defenses.
Okay, nervous, endocrine, immune. Any other major players?
Oh, yes. The list goes on. We also have retinoids, which come from vitamin A, and then there are the eicosanoids. Eicosanoids like prostaglandins and leukotrienes are really key players in inflammation, usually acting locally. They're derived from arachidonic acid. And we can't forget growth factors. These are polypeptides that tell cells to divide or just get bigger.
Wow, okay. So many different kinds of messengers. It really is an elaborate language. But how does a cell actually read these messages? It doesn't have eyes or ears.
Yeah,
Right?
that's where the receptors come into play. Receptors are these specialized proteins, usually found either on the surface or inside a target cell. They are incredibly specific. Think of them like a lock that only fits a very specific messenger key. Once that messenger binds, it kicks off something called signal transduction.
Signal transduction. Okay, what exactly is that?
That's the crucial step, really. It's the process of converting that external message, the messenger binding to the receptor, into an internal response within the cell. It's the translation step, you could say, that leads to actual changes in the cell's behavior.
Got it. Translation. And you mentioned receptors can be in two main places, inside the cell or on the surface. Why the two different locations? What determines that?
Yeah, that's a great question. It boils down to the chemical nature of the messenger molecule itself. Is it fatty or watery, basically? If a messenger is hydrophobic, meaning it's fat-loving, it can easily slip right through the cell's fatty outer membrane, the plasma membrane. These types of messengers bind to intracellular receptors, which are found floating around inside the cell cytoplasm or maybe in the nucleus. Steroid hormones, like cortisol, work this way.
Okay, so they just walk right in?
Pretty much. But if a messenger is polar or water-loving, it can't easily cross that fatty membrane barrier. For these guys, the cell needs, well, a doorbell on the outside, and that's the plasma membrane receptors. They sit on the cell surface. Peptide hormones and most neurotransmitters use these surface receptors.
Makes sense. Different messengers need different ways to get the message across the boundary.
Exactly. And this fundamental difference, whether the receptor is inside or outside, dictates the very first steps of the whole signaling cascade that follows. But regardless of the type, there's a kind of universal sequence to it all. First, the messenger is secreted. Then it's transported somehow to the target cell. Then the messenger binds its specific receptor. That binding elicits an intracellular response. And finally, crucially, the signal gets terminated, turned off.
And only the cells with the right lock, the right receptor, will actually hear that specific message.
Precisely. Specificity is key.
Okay, let's talk about those intracellular receptors first, the ones for messengers that slip inside. What kind of changes do they trigger? Are we talking quick fixes or more like long-term adjustments?
Generally, these are about the long game. Most intracellular receptors are actually powerful gene-specific transcription factors. So when a hydrophobic messenger, like a steroid hormone, gets inside and binds its receptor, the whole complex messenger plus receptor often moves right into the nucleus if it wasn't there already. Once in the nucleus, it binds to specific DNA sequences. These are called hormone response elements. Think of them as specific docking sites on the genes. This binding directly regulates the transcription of certain genes, essentially turning them up or down. It changes what proteins the cell makes.
Whoa, so these signals aren't just tweaking current activity, they're literally reprogramming the cell's blueprint for the longer term. That sounds incredibly powerful.
It is powerful, and it leads to these significant adaptive responses.
Can you give us an example of that in action?
Sure. A classic one is cortisol action. Cortisol, the stress hormone, travels through the blood, often bound to transport proteins. When it reaches a target cell, say in the liver, it diffuses inside and binds to its specific intracellular glucocorticoid receptor. This binding causes the receptor to change shape, allowing the whole complex to enter the nucleus and bind to specific DNA sites called glucocorticoid response elements. The result: increased production of certain enzymes, like those involved in gluconeogenesis. That's the process of making new glucose.
Gluconeogenesis, making new sugar.
Right, and we can see this clinically. Take the case mentioned in Marks and R, who suffered from anorexia nervosa. Her prolonged fasting and a chronic exercise meant her body was under constant stress, leading to high cortisol levels. This cortisol, acting through these intracellular receptors in her liver, ramped up the production of those gluconeogenic enzymes. It was her body's way of preparing to synthesize more glucose to cope with dangerously low fuel reserves. It's a vital survival mechanism, though it's a slower response compared to some others.
That makes total sense. A slow, profound adaptation to a chronic state. Okay, now let's shift focus to the doorbells on the cell surface, those plasma membrane receptors. What do they generally look like, and how do they handle their signals?
Right. All plasma membrane receptors share a sort of basic structure. They have an external part, the domain that actually binds the messenger molecule. They have one or more segments that span right through the cell membrane, usually alpha helices. And they have an internal part, the intracellular domain, that kicks off the signal transduction cascade inside the cell.
So outside, middle, inside.
Pretty much. When the messenger binds to that external domain, it causes a shape change, a conformational change, that ripples through the receptor to the inside part. And that's what starts the internal signal. These pathways can trigger really rapid effects, changing ion levels across the membrane, or flipping enzyme switches on or off almost instantly. Or they can lead to slower, more sustained effects, like altering gene expression, similar in outcome to intracellular receptors, but through a different route. Often, actually, you get a mix of both fast and slow effects from the same signal.
Okay, and these surface receptors fall into different categories too, right?
Yes, they generally fall into three major classes based on how they initiate that internal signal. There are ion channel receptors, receptors that are kinases or bind kinases, and the very common G protein-coupled receptors.
Let's start with those ion channel receptors. They sound like they'd be involved in really fast processes.
They absolutely are, built for speed. When a messenger binds to one of these, it directly causes an ion channel, which is part of the receptor protein itself, to open or sometimes close. This allows specific ions like sodium Na+ or potassium K+ to flow rapidly across the membrane down their concentration gradients. This rapid ion movement can generate an electrical signal, like in nerve cells, or trigger some other very quick cellular response.
Can you give us the classic example?
The nicotinic acetylcholine ACSH receptor is the textbook example. ACA is a neurotransmitter released by nerves, particularly at the junction between a nerve and a skeletal muscle cell. It diffuses across that tiny gap, the synapse, and binds to these nicotinic AC receptors on the muscle cell membrane. That binding instantly opens the channel. Sodium ions rush in, potassium ions flow out, and this rapid change in ion flux initiates the electrical signal that ultimately causes the muscle cell to contract. It happens in milliseconds.
Lightning fast. That makes sense for muscle control. But you also said this highlights how delicate the system is. If that switch, the receptor, is broken,
Then the whole circuit fails. And this leads us to a really compelling clinical connection: myasthenia gravis. Think about Mia S., the patient described in the text. She experiences debilitating muscle fatigue, especially, say, when she's chewing or talking for a while. What's happening is that her own immune system is mistakenly attacking and destroying her nicotinic AC receptors in her skeletal muscles.
an autoimmune attack on the receptors themselves.
Exactly. So with fewer functional receptors, even when her nerves release AC correctly, the muscle just can't respond effectively. The signal transmission is impaired, especially with repeated effort when AC levels might fluctuate or receptors become temporarily less responsive.
Wow. And that explains why certain drugs might help her.
Precisely. A drug like adrophonium temporarily inhibits the enzyme, acetylcholinesterase, that normally breaks down AC in the synapse. By slowing down AC breakdown, more AC hangs around in the gap for longer, increasing the chances it will find and activate one of the few remaining functional receptors. This can briefly improve her muscle strength, which is actually used as a diagnostic test. It perfectly illustrates how critical those receptors are.
That's a fascinating link between the molecular mechanism and the patient's symptoms. Okay, let's move to the next class, the receptors that are kinases or bind kinases. That sounds like it involves maybe adding phosphate groups.
You got it. Phosphorylation adding phosphate groups is central here. These receptors either are protein kinases themselves, meaning they have the enzyme activity built in, or they associate very closely with separate protein kinases floating in the cytoplasm. When the messenger binds, it activates this kinase activity. The kinase then adds phosphate groups, PO4, onto the specific amino acid residues, commonly tyrosine, serine, or threonine, on the receptor itself. That's called autophosphorylation, or on other target proteins inside the cell.
Like adding little molecular on switches.
Exactly. It's like adding a sticky phosphate tag that changes the protein's shape or activity, or creates a docking site for other proteins to bind. This often initiates a chain reaction, a phosphorylation cascade, where one activated kinase phosphorylates and activates the next kinase, and so on, amplifying the signal as it goes deeper into the cell.
And a key example here involves insulin, right?
Yes, the insulin receptor is a prime example of a receptor tyrosine kinase. It actually exists as two halves, monomers, that come together, they dimerize, when insulin binds. This dimerization activates their intrinsic tyrosine kinase activity. They phosphorylate each other on specific tyrosine residues. These newly phosphorylated tyrosines then act like docking stations, attracting other signaling proteins that have specific domains, often called SHU domains, which recognize these phosphotyrosines.
So the receptor becomes a kind of scaffold once it's activated.
A scaffold or a hub, exactly. One key protein that docks is called insulin receptor substrate, or IRS. Once IRS binds and gets phosphorylated by the insulin receptor, it becomes a hub, recruiting even more proteins. This allows the initial insulin signal to branch out, to diverge into multiple downstream pathways. One major pathway involves activating RAS, a small G protein, which then triggers the MAP kinase cascade, RAF-MAPKK-MAPK. This pathway is heavily involved in regulating gene expression related to cell growth and survival.
Okay, so one branch affects genes. What else does insulin do?
Another critical branch involves activating PI3 kinase. This enzyme phosphorylates specific lipids in the cell membrane, creating new docking sites right there on the membrane surface. These new lipid docking sites recruit other kinases, like protein kinase B, also known as ACT, The PI3 kinase ACT pathway is crucial for many of insulin's metabolic effects, like stimulating glucose uptake into muscle and fat cells, promoting glycogen synthesis in the liver and muscle, and also contributing to cell growth and survival.
So it's like a molecular relay race, but the runners can branch off onto different tracks, leading to diverse outcomes like glucose uptake and gene changes.
That's a good way to think about it. And it helps explain the problems seen in diabetes. Consider Diane A., the patient with type 1 diabetes. She lacks insulin entirely. So her insulin receptors are never activated. This means glucose doesn't get transported properly into her cells, leading to high blood sugar, hyperglycemia. And the pathways promoting glycogen storage and muscle protein synthesis are also shut down, which contributes to the muscle wasting sometimes seen in uncontrolled diabetes. It really highlights how one messenger, through these kinase cascades, coordinates multiple vital functions.
It really does. Are there other important kinase-linked receptors?
Oh, absolutely. There are also tyrosine kinase-associated receptors. These receptors don't have their own kinase activity, but they recruit cytoplasmic tyrosine kinases, often from the Jake family. Cytokine receptors work this way. Binding of a cytokine brings the receptor parts together, activating the associated J kinases, which then phosphorylate the receptor and downstream proteins called STATs. These phosphorylated STATs then travel to the nucleus to regulate immune response genes.
Jason's stats. Okay, another pathway.
And then there are receptors with intrinsic serine threonine kinase activity, like the TGF-β receptors. They phosphorylate downstream proteins called SMADs, which regulate genes involved in things like tissue repair and development. So kinases are everywhere in signaling.
It seems like adding and removing those phosphate groups is just a fundamental way cells flip switches. Now let's get to the big one, the most common type you mentioned, the heptahelical receptors, or G-protein-coupled receptors, GPCRs. What makes them so special and so widespread?
Right, the G-protein-coupled receptors, or GPCRs, they are incredibly versatile, and yes, they are the largest family of cell surface receptors we know of. They all share this characteristic structure of having 7 segments that snake back and forth across the cell membrane, hence heptahelical. Unlike the kinase receptors we just talked about, GPCRs don't have their own built-in enzyme activity. Instead, their intracellular domain interacts with a separate group of proteins called heterotrimeric G proteins.
Heterotrimeric.
Yeah.
Meaning 3 different parts.
Exactly. These G proteins are composed of 3 different subunits: alpha, beta, and gamma. In the inactive state, the alpha subunit has a molecule called GDP bound to it. When a messenger binds to the GPCR on the outside, the receptor changes shape. This shape change is transmitted to the associated G protein on the inside. The GPCR then acts like a catalyst, prompting the G-alpha subunit to release its GDP and bind a molecule of GTP instead.
So binding GTP flicks the switch to on for the G-alpha subunit.
Precisely. Binding GTP activates the G-alpha subunit. It then typically dissociates from the beta-gamma subunits and goes off to interact with, and usually activate, a specific target protein nearby in the membrane. This target protein is often an enzyme or sometimes an ion channel.
Okay, so the GPCR activates the G protein, which activates a target enzyme, and then what? This is where second messengers come in.
Yes, this is where the amplification often happens via second messengers. The activated target enzyme then starts churning out large quantities of these small, non-protein intracellular signaling molecules. The original hormone or neurotransmitter is the first messenger. These small molecules made inside are the second messengers. Common examples include cyclic AMP, NTAP, inositol trisphosphate, IP3, diacylglycerol, DAG, and even calcium ions, Ca2+.
And these second messengers spread the signal far and wide within the cell.
They do. They diffuse rapidly throughout the cytoplasm, activating downstream targets, often protein kinases, leading to a much larger and broader cellular response than you'd get from just the initial messenger binding event. It's a major amplification step.
Can we walk through a key example pathway, maybe CMP?
Sure. The CMP pathway is a classic. Some G proteins have what's called a G-alpha subunit, as for stimulatory. When activated by its GPCR, ASIAS activates the membrane enzyme adenylyl cyclase. Adenylyl cyclase then takes ATP, the cell's energy currency, and converts it into cyclic AMP, CMP. CMP levels rise rapidly inside the cell. This GAMP then binds to and activates protein kinase A, PKA.
PKA, you know the kinase.
Yes. Kinases are central. Activated PKA then goes on to phosphorylate a whole range of target proteins, including enzymes involved in metabolism. For instance, the hormone glucagon acts via this pathway in the liver to stimulate glucose production when blood sugar is low. That's relevant to ANR. Again, activating glycogen breakdown and gluconeogenesis via PKA phosphorylation. cIMP can even move into the nucleus and affect gene transcription by activating a protein called CRE.
So CAMP does a lot. What about the other second messengers you mentioned, IP3 and DAG?
Right, that's another major pathway activated by different GPCRs, often those coupled to a G-alpha-Q subunit. GAC activates a different membrane enzyme called phospholipase P, PLC. PLC cleaves a specific lipid molecule in the plasma membrane, called PIP2, into two different second messengers simultaneously: inositol trisphosphate, IP3, and diacylglycerol, DAG.
Two for the price of one. What do?
IP3 is small and water-soluble, so it diffuses into the cytoplasm and binds to receptors on the endoplasmic reticulum, causing stored calcium ions, CO2+, to be released into the cytosol. Calcium itself then acts as another important second messenger. DAG, meanwhile, stays associated with the plasma membrane because it's lipid-like. There, along with the increased calcium, it activates another kinase called protein kinase C, PKC, which then phosphorylates its own set of target proteins.
Wow, that's quite the cascade. GPCR activates G protein, activates PLC, makes IP3 and DAGA. IP3 releases calcium, DAGA and calcium activate PKC. A lot of steps.
It is intricate, but it allows for very fine-tuned and diverse responses. But, one of the most elegant parts of this G protein system is how it turns itself off.
Right, because these powerful signals can't just stay on forever. How does that work?
Well, the G-alpha subunit has a built-in feature, an intrinsic GTPase activity. This means the alpha subunit itself can slowly hydrolyze the bound GTP back to GDP.
It has its own off-switch timer.
Exactly. Once GTP is hydrolyzed back to GDP, the G-alpha subunit becomes inactive again, dissociates from its target enzyme, and reassociates with the beta-gamma subunit, ready for the next signal. It's an automatic reset mechanism.
That's incredibly clever, a self-limiting switch.
It is, and the importance of this internal clock is dramatically highlighted when it gets broken. This connects back to the bigger picture. The versatility is amazing, but disrupting the control is dangerous.
And this brings us back to that devastating example you mentioned earlier, cholera.
Yes, precisely. Dennis Faith, the patient with cholera, described in Marks. The cholera toxin, produced by the bacterium Vibrio cholerae, has a subunit that gets into intestinal cells. What this toxin does is it chemically modifies the GIL subunit, the one that activates adenyl cyclase. Specifically, it locks it in the GTP-bound active state by completely disabling its intrinsic GTPase activity. It breaks the off switch.
So the GELOS is just stuck on?
Permanently on. This means adenyl cyclase is constantly being stimulated, churning out massive amounts of KMP. In intestinal cells, this huge surge in KMP leads to the overactivation of a specific chloride channel called CFTR. This causes a massive efflux of chloride ions, sodium ions follow, and water follows osmotically out of the cells and into the intestinal lumen,
leading to that severe watery diarrhea and dehydration characteristic of cholera.
Exactly. It's a terrible disease, but it's a powerful illustration of what happens when just one component of signal termination, that G protein GTPase activity, fails.
Wow, a broken off switch with devastating consequences. You also briefly mentioned cGMP. Is that similar to CAMP?
It is, in principle. There are receptors that are guanylyl cyclases, converting GTP to cyclic GMP, cGMP. And cGMP acts as a second messenger, typically activating protein kinase G, PKG. Interestingly, there's also a soluble form of guanylyl cyclase inside cells that acts as a receptor for nitric oxide, NO, a gas that can diffuse across membranes. No signaling via cGMP is important in relaxing smooth muscle, for instance.
And drugs can target this?
Yes. Drugs like sildenafil, used for erectile dysfunction, work by inhibiting the enzyme, a phosphodiesterase, that normally breaks down cGMP. By keeping cGMP levels higher for longer, they enhance the signaling pathway that leads to smooth muscle relaxation and increased blood flow.
Clinical relevance again. And you mentioned eicosanoids earlier in the context of inflammation.
Right, like with Lotta T and her gout. In gout, those sharp urate crystals trigger inflammatory cells to produce and release leukotrienes, which are a type of eicosanoid derived from arachidonic acid. These leukotrienes then act in a paracrine fashion locally on nearby nerve endings and blood vessels, stimulating pain receptors and contributing to the inflammation and intense pain of a gout attack.
So it really covers everything: lightning fast, muscle twitches, long-term gene changes, metabolic shifts, immune responses, inflammation, even diseases like cholera and conditions like gout. It all comes down to these tiny, precise molecular conversations. But we've touched on it with the G proteins, how crucial it is to stop the signal. How else does the cell make sure these conversations don't just echo on forever? How do they achieve signal termination generally?
That's such a critical point. Saying stop is just as important as saying go in cell signaling. If signals aren't terminated properly and promptly, you can get uncontrolled cell growth, like in cancer or other serious diseases. The timing matters too. Some signals need to be shut off in milliseconds, others can fade more slowly. Cells use a whole toolkit of strategies, not just the G protein GT-PACE activity.
Okay, what else is in the toolkit?
Well, one obvious way is to get rid of the messenger itself. Messenger degradation. Enzymes can rapidly break down the signaling molecule. We saw that with acetylcholinesterase, quickly chewing up AFT at the synapse. Another is second messenger inactivation. Just as enzymes make second messengers, other enzymes degrade them. For example, CMP phosphatidylinosyl breaks down PPP into inactive AMP, shutting down the CKP signal. We just mentioned drugs that inhibit this enzyme.
So degrade the first messenger or the second messenger. Makes sense.
Then there's the G protein's GT-PACE activity, that internal clock we discussed. And this can actually be sped up by other regulatory proteins called GT-PACE activating proteins, or GAPs, providing another layer of control.
Okay, turning off the G protein switch faster.
Cells can also target the receptors themselves through receptor desensitization and downregulation. Receptors can be chemically modified, often by phosphorylation, which makes them less responsive to the messenger or unable to activate downstream pathways, even if the messenger is still bound. That's desensitization. Or the cell can physically remove receptors from the surface through endocytosis, pulling them inside the cell. These internalized receptors might be recycled back to the surface later, or they might be sent off to be destroyed in lysosomes. This downregulation reduces the number of available receptors, making the cell less sensitive to the signal overall.
So making the existing receptors deaf, or just getting rid of them altogether.
Essentially, yes. And finally, remember all those phosphorylation events we talked about with kinases? There's a whole army of enzymes called protein phosphatases, whose job is to do the opposite. They remove those phosphate groups from proteins. By dephosphorylating the components of signaling pathways, phosphatases effectively reset the switches, turning off the signals that were initiated by kinases. It's a constant balance between kinase activity adding phosphates and phosphates activity removing them.
Kinases put them on, phosphatases take them off. A dynamic equilibrium.
Precisely. And again, if these termination mechanisms fail, if phosphatases don't work right, or receptors aren't downregulated properly, or messengers hang around too long, the consequences can be profound. We saw it dramatically with cholera, but it's a factor in countless chronic diseases. Maintaining that balance between on and off is fundamental to health.
Absolutely. What an incredibly intricate yet really elegant system for coordination and control. So let's try and bring this all together for you, our listener. Today we've really journeyed deep into the world of cell signaling by chemical messengers. We've seen this amazing cast of characters, neurotransmitters for those rapid nerve signals, hormones traveling long distances for systemic effects, cytokines coordinating immune responses, and others. They all orchestrate almost everything your body does. We explored the sophisticated receivers, the receptors that cells use to hear these messages. We saw how they fall into two main camps. The intracellular receptors for fat-soluble messengers like cortisol, which often go right to the DNA to change gene expression long-term, and then the diverse plasma membrane receptors for signals that can't get inside easily. These included the superfast ion channels, like the ACEP receptor, and we heard how its destruction causes myasthenia gravis. We delved into the complex world of kinase-linked receptors, like the insulin receptor using phosphorylation cascades to control metabolism and growth, and how defects here lead to conditions like type 1 diabetes. And we can't forget the most common type, the versatile G-protein-coupled receptors, or GPCRs. They use G proteins as switches to trigger powerful second messenger systems, like CMP and IP3DG, amplifying signals enormously. And the cholera example showed us just how critical the GPCR off switch really is. Crucially, threaded through all of this, we learned that turning the signal off is just as vital as turning it on, using a whole array of mechanisms from messenger degradation to receptor removal and dephosphorylation. So if we had to boil it down, here are the main takeaways for you from our discussion.
Yeah, first, cells are constantly communicating using this huge variety of chemical messengers. This communication integrates and drives pretty much all bodily functions.
Second, those receptors are highly specific listening devices. Their location inside the cell or on the surface and type determine exactly how a cell hears and responds to any given message.
Third, these signal transduction pathways, they're often intricate cascades involving kinases, phosphatases, G proteins, and second messengers. They're the mechanisms that translate the external signal into real internal action.
And fourth, both starting the signal and carefully stopping it are absolutely vital for your health. Breakdowns in either initiation or termination are really at the heart of many, many diseases, from autoimmune conditions like myasthenia gravis to infections like cholera and even complex diseases like cancer and diabetes. So as you go about your day, maybe just take a moment to consider this silent, unseen symphony of chemical signals constantly happening within you. Every thought you have, every move you make, even just breathing, it's all a testament to this incredible cellular communication network. And a provocative thought to leave you with. We've talked about these pathways, but scientists are increasingly understanding the crosstalk, the intersections between these different signaling routes. What new discoveries about how these pathways interact and influence each other might further revolutionize our understanding of health, disease, and maybe even medicine in the years to come. We really hope this exploration has given you a new appreciation for the just amazing biochemical choreography happening inside you every single second. Keep exploring, keep questioning, and thanks for diving deep with us today.